Um, Before I start, I'd like to thank the session chair for letting us present this. And I say us because even more than normal single omic science, once you're doing multi-omic studies, you're going to be dealing with multidisciplinary teams. In our case, we had MDs and microbiologists. We had proteomics experts, metabolomics experts, and bioinformaticists all working together to try and make a coherent picture in the study of Staph aureus in clinical isolates. And in fact, this is such a multi-type setting that it's also, and we've seen this in some of the other talks, it's multi-site as well. So I'd like to acknowledge everybody up front because it's always a group effort. Now, the other thing is I want to apologize in advance because it's going to be a slightly different kind of talk. I'm going to only look at 2 ohms, and I'm going to try and use one tool and go in detail. So this is a bit of a data jam, which means I'm going to be talking very quickly. But you guys have you people have chosen to be here rather than outside in the San Diego sun. So you're obviously up for it, and so let's get started. The setting for this is we're going to be looking at MRSA, which is Staph aureus that's antibiotic resistant, and in the press you'll see it referred to as a superbug. Very scary stuff. It definitely kills patients. It also, if you follow this sort of thing, causes damage to the economy. Usually it's thought of as healthcare associated or hospital acquired, and you may be thinking, yes, but I am fit, I go to the gym. Well, there's a MRSA for you as well. There is community-acquired MRSA. So we all have skin in this game, and we should understand the virulence of this stuff. Now, to understand the virulence, to understand anything, any quantity, you need to be able to measure it. So in our case, when we talk about virulence, we're going to be talking about cytotoxic virulence. And the way you measure this is you grow the bugs, you collect the supernatant. It has a bunch of virulence factors that it's secreting. You pour that on poor, unsuspecting neutrophils. They die for science. You measure how much they die. That's your measure of virulence, okay? And that's what we're going to be studying. So anytime you see virulence on a plot, this is what was measured. That's the sort of phenotype. Now, if you, maybe the main, the first takeaway is that genomics is not enough. So what you're seeing here is a tree of strains that are bloodstream isolates. So these are from very sick patients, and we're clustering them. And what you're seeing is some of the, well, you can't see it, I'm going to tell you. Some of these strains are low virulence. Some of these strains are high virulence. And it's not that they cluster, obviously, by virulence. And you might not expect that at a genomic level, at a gross genomic level. But even if you unpack and look at the SNPs, et cetera, you can't make a predictor for virulence just from sequence. And yet the difference is pretty profound in the ability of these guys to kill. So this, we need something more, right? Now, proteomics can obviously help. So here we're clustering data by proteomics, and that tracks cytotoxicity quite well. And it's not that surprising because we're measuring the tools of the bug's trade. So it's making cytotoxins, we're measuring them. We're measuring other virulence factors that are known to be associated with cytotoxicity, sometimes inversely correlated, as you can see, but basically we're already dealing with the actual tools of the trade of the bug. Now, what's maybe less obvious is that metabolomics is much more important here than you might suspect initially. So if you take one of these strains, it's listed here as having 1 cytotoxicity level. But if you modulate the amount of some metabolites in the growth media, it has profound effects on the cytotoxicity of these bugs. So it's important to measure proteomics and metabolomics, and therefore Victor Torres put together a team of mass spectrometrists specializing both in the measurement of proteomics and metabolomics. And they are also very conveniently located smack dab in the middle of a bunch of hospitals in Manhattan. And we have allied MDs who are experts at finding strains that are troubling and bringing them into a collection. So this is a sort of an ideal setting to do proteometabolomics of this bug. Now, if you're going to do proteometabolomic analysis, you're going to need to have some sort of data system to project the data onto. And so you're going to need pathway information, and you're going to get it from various standard sources. Now, one of the things that I want to suggest here is that you want to curate this yourself. So you want to get these you want to set up a database of reactions that you're getting from the literature, but you want to curate it yourself for two reasons. The first of which is just knowing what's in the database will allow you to know what questions you can even answer, because if there's no link from A to B, you won't be able to get data that shows it, certainly not causally. You need that network to be in place. And the other thing is, this is indeed a causal network. This is the original causal network. We are saying here, this enzyme causes these substrate to turn into these products. So working around a well-defined, well-understood causal network is very important. And if you do build such a biochemical reaction network, please use a graph database and not a relational database. This is an aside for the bioinformaticists in the audience. And it's also a well-understood choice now. Reactome uses it. This is a very good reference for a graph database. So do make your own database, and I suggest using a graph database to do it. Now you're going to collect your sample. You're going to separate it as late as possible into proteomic and metabolomic, and you're going to project that onto your network. And I apologize for the ridiculous oversimplification of how that's done. My expertise is from the raw file onwards. I have no idea how the prior steps work in detail. I don't know anything about separation science, so I'm simplifying that criminally. But the fact of the matter is that we took these 8 strains and ran them through a proteometabolomic characterization. And then we got a spreadsheet. That spreadsheet has 3 tabs. The first tab is proteometabolomic, so it's organized by reaction. I don't know if you can see the color difference. So you have the metabolites and the proteins per reaction. You have a tab just for the proteomics. You have a tab just for the metabolomics, right? So on paper, we're done. We did our job. We gave a proteometabolomics characterization of these strains. But of course, we're not done, because you want to see what the heck you're looking at. I'm sorry about my language. And there are many ways to fail at this. Failure number 1 is you make endless lists. You show dense hairballs, or you lean too heavily on pre-computed canonical views of the data. So endless lists, I just showed you. You show just endless lists of data. The other one is dense hairballs. You see a lot of this in systems biology because it allows us to show the amazing amount of data that we've collected. But this is incredibly misleading. So for example, here, those 8 networks are showing exactly the same information. They look completely different because it's all about the layout. And so we have way too many hairballs in systems biology, and we have to stop it with the hairballs. Okay, so no more hairballs. Right? Now, a subtler way to fail at this is you lean too much on the framed way of looking at things that comes in the encyclopedia, in your KEGG and your whatever. So there are canonical pathway maps, and you project some of your data just on that individual window, and you might miss action that's on the boundary of 2 frames, right? So we want to project and look at everything, but as I mentioned earlier, you don't want hairballs. So what are we going to do? We're going to try and find a hotspot in the network and focus just on that region of the biochemical network. And so what I mean by a hotspot is an area where there's change. This has been done before, particularly the transcriptomics people have tools for this. The network that they're usually doing this on is regulatory networks, genetic networks. But the framework is similar. So this is not a crazy novel idea, but you don't see it a lot in proteometabolomics. Now, just a few definitions. By heat, and I'm sorry to abuse thermodynamics here, heat here just means change. You could use p-values. We prefer to use log of fold change in absolute because we don't care about the direction. If you're up or down, something interesting is going on. We like this measure because it speaks to the end user. P-values are not what we usually deal with when we're reasoning about networks. We're reasoning about what changed up or down. Now, we're going to be looking per reaction at the average change, difference, heat, of the proteins in the reaction or the metabolites in the reactions. And the reason we keep those separate is that they have very different topological profile in the networks. In particular, metabolites are present. Sometimes there are subsets of metabolites that are present in a bunch of reactions, and that doesn't happen in proteomics. So you need to treat the data subtly differently. But conceptually, you're just tracking heat in these reactions. But for example, for the metabolites, we're going to ignore currents, what are sometimes called currency metabolites in the analysis, ATP, et cetera. You don't want to find links in things that are coming through basically very common currency metabolites. Now, what you then do is you combine your proteomic and your metabolomic heat into local heat for one reaction. And then the next two steps I'm going to show graphically, so don't be alarmed. But the first thing we're going to do that is very important is we're going to do a diffusion step. So we're going to allow heat from neighboring reactions to flow in the biochemical reaction network. And the reason is sometimes you can't measure everything, but all of your neighboring reactions are hot, and you don't want to drop a reaction just because you weren't able to cover it. Then what you do is you drop the things that don't have any heat at all, and you find components. So just graphically, I just want to show you what that looks like. Graphically, we have reactions. They're connected. If one produces something, the other consumes. That's pretty straightforward. We have a diffusion step. So some reactions will borrow heat from their neighbors. You remove the ones that don't, and what you're left with are hotspots. And you usually go straight for the biggest one because we're super curious, but you technically are generating a bunch of hotspots. Now, it's important to point out that when you do this kind of analysis, all you need really are canonical accessions and ratios, and you're off to the races. So you don't need too much, and the database of reactions that we talked about earlier. So I'm going to try and show you what this looks like. So we're doing a little bit of a data jam here, and I'm going to be speaking very quickly, so I apologize for that, but let's take a look. So first of all, you're seeing I just violated my own no hairball rule. I'm showing you a hairball, but it's just for context. That's all of our data. Now we're going to take some parameterization, thresholds that I mentioned earlier with the equations, and we're going to get a hotspot. And so a hotspot is a subset of the network, and we're going to show graphically where there seems to be action in the network. So if I just zoom in here on one of the reactions, the halo around the reaction says there is something going on. And you can see that there are other reactions that have less to no measurements done at all on them, right? Now, we really want to see what is changing. So we expand the node, and we can see the substrate, the enzymes, and the product per column. And if you forget structures, you click a button, you can see the structures. If you forget all of the textbook information about Pure C, you can go to UniProt. So there are link outs to the relevant information, but basically you're seeing the quantitative information to the extent that it's available. And the only downside of this view is that some quantitative measurements are overrepresented because they occur in more than one reaction. So we have another view, which is a little jarring, so I apologize for this, where everything is only represented once. So it's a little jarring, and it's a little bit more hairballish, but I just want to show you in principle what that looks like. And now every measurement that we ever made is only represented once, except you'll see in a second for the currency metabolites. Otherwise, you get a horrible mess again. I mean, arguably, this is already a horrible mess. But so you can see aspartite is here only once. If we look at any one of these reactions, you can see the substrates, the product, and the enzyme that catalyzes the reaction. We show that explicitly because there may be more than one, by the way. And importantly, the things in dashed lines are metabolites that are currency. If we were to represent them only once, we would again collapse into a terrible hairball. So whatever the visualization that you use, you need to take some care into what you're showing so that it's somehow mappable. But I would argue this is still a hairball. It's not my favorite view at all. So let's collapse that down and go back to a view where you see all of the measured information per reaction. And what you're seeing here, even at a glance, is that the hot reactions are hot because of a down regulation in the proteins, which is the middle column. That's why they're all blue. And you would like to arrange that a little bit by some prior knowledge, but without bringing in a full-blown pathway. So we have this notion of a backbone. And a backbone is a walk through a pathway. So it's less than a pathway. So we get a backbone assist. We ask the system, is there any backbone that's represented here? What's the largest backbone represented? And we find in this case, and these by the way, come in our case from something that Keg calls Keg modules. And it suggests sets of reactions that can be seen as a path. And that's why we call it a backbone, because it looks like a backbone. Personally, I prefer a backplane, so we arrange things in a backplane. And you can immediately see, by the way, the benefit of the diffusion step, because some of the participants in this path had no measured data, but they still made it here into the hotspot thanks to their neighbors. And now what's left is to basically annotate around this backbone. So there's a huge back loop that's not normally represented in this, which is interesting. That's the glycine source. I'm just rearranging things a little bit. This is an alternative pure N step, so the backbone chose one version of the reaction rather than another. And so what we're going to do here is we're going to say there's actually duplication of the description of what's going on here. So we're going to delete one of these reactions, which we don't have any empirical evidence for either of these anyway, so let's just remove one and the associated sort of pre-processing step, if you will, here. Okay, so we've simplified that. There's two more reactions left in the purine biosynthetic pathway, this one that I'm moving right now, and the next one. Everything else is off the pathway. You wouldn't see it in the textbook. And so we're just going to lay it out a little bit, and I just want to show you. Sometimes you'll see suggested enzymatic steps that look like reverse reactions. But if you're a chemist and you know what you're doing, you can click on these and see that they're subtly different. So you always have a reminder of the structures involved as well. Now, all of the adjacent reactions are suggested by the metabolomic data primarily. So I just want to very quickly show you one important little detail about tools such as these. Any tool that tries to summarize the data should always leave you a way to get back to the underlying measurements. So here we're going to just double check that we do have an upregulation of glycine. So this is the actual mass spectrometry data. And then one final observation that I want to make is I want to show you an imperfect result, because sometimes those are the most interesting. I just want to show you that sometimes you have an upregulation that if you filtered by p-value, you wouldn't be able to see, because it has what looks like an error. There's a red line that should be blue and a blue line that should be red. And you would think maybe some vials were swapped, but they weren't. What actually happened is the subject of a whole talk in and of itself, but you need the ability to go back to the underlying data. So because I overran, I just want to say, I hope I've convinced you that it is possible. Oh, just one final thing. The fact that purine biosynthesis is associated with virulence is a matter of hot research right now, but it's usually in strains that have been generated by screens, by mutagenesis. This is in the wild, so that's the main biological result. And I hope I've been able to show you that using this kind of tool, we can get to the relevant area of the biochemical network, and we're not limiting the user to predefined boxes. And obviously, this can be done for other organisms, even single omics data sets, if you're still in that mode. Or, heaven forbid, you're doing transcriptomics and you're not using a mass spec. Shame on you, but you can use this too. There you go. And that's, sorry I overran a little bit. Thank you.
This is the tail end, but if there are questions, please, we would welcome them. In the meantime, is the software available and open source, or how are you distributing it?
So what I'm doing right now is I'm collecting interest. Believe it or not, when you propose to do this, I mean, people in this session, it's obvious to you guys that this is useful, but it's not entirely obvious out there. Oh, why would you do multiple ohms? So what we're doing right now is we're collecting a list of people who would like to try this, and then that's how we'll decide how to. But yeah, the whole point of this is to make a useful tool. Absolutely. Yeah.
So very nice. I enjoyed the talk very much. I have a couple of questions. One of them is about the local heat that you mentioned. So you're limited by having both protein and metabolite information in order to generate that, right? So for each of the hotspots, for each of those nodes, you're going to have a value. You have to have a value for both. Because if your data is sparse on the metabolites, it's going to be an issue.
So in fact, you can run the tool in two modes. In one mode, you have an interaction term forcing there to be evidence from both ohms. But in another mode, which is the mode I showed here, if there's enough evidence from one mode, there'll be enough heat to be in the network. We've run data sets with just 1 ohm.
With just 1 ohm.
In fact, the tool has, if you saw, there's a slider. You can take a multi-ohmic data set and pretend you only had the metabolites or only had the proteins.
Great.
That helps you to see where each technology sends you, actually.
And the other question is about the ratio. So you're doing taking the log base 2 of the ratio. So that's nice. You don't dealing with p-values. What do you do when the actual abundances that you have are very low?
Yeah, so you're right that low abundance measurements are going to generate wild ratios, and that can affect things. So this is let's open this up a little bit. I like to use ratios because they speak to people. I pay a price. The price is you have to filter the data sets so that you're dealing with measurements that are reliable. Okay. But obviously you can modify this to pre-filter by p-value. There's all sorts of ways you could do this. The key is to have a data system where you can project all of this and then do that hotspot analysis. But you're right, the low intensity measurements can generate wild ratios.
Thank you. Teo, come on up if you wish to continue. There was another question? Okay. In that case, listen, all of you, thank you for staying to the end. I hope it was worth it. I think it was worth it. Thank you very much. See you next year.